Hi, everyone. Welcome to the Johns Hopkins seminar series on the cerebellum. Today, as our guest, we have Ming-Kai Pan. Ming-Kai received both his PhD and MD from the National Taiwan University, where he's now associate professor and an attending physician. He's a movement disorder specialist whose work has significantly advanced our understanding of the pathophysiology of essential tremor. For example, his work has identified a pathophysiological contribution to tremor beginning at the structural level, climbing fiber to Purkinje cell synapses, all the way to the behavioral level, kinematics of tremor. Additionally, he has made significant contributions to bioengineering, particularly in the development of novel motion tracking technologies and volumetric supersede microscopy. Ming-Kai, thank you so much for presenting for us.
Thank you so much for that. And then it's my great pleasure and great honor to be here and to share our recent discoveries in terms of motor control. I have several disclosures that work with some as a consultant in drug companies, but there's nothing to do with today's talk. So before I start, then let me talk briefly about how I'm doing. So as you know that you're pretty much familiar with the field of neurodynamics, live neural recording in mouse and humans. So as a physician, I had the privilege to record a lot of human cerebellar EEGs. So we are recording this EEG in the cerebellar field and non-invasively, so to understand how they control their emotions. And we are also able to do this cognitive-based EEG recordings in computer-based cognitive tasks. So I'm recording how their cerebella and their whole brain react to their during their cognitive process. And we are able to use some music-based super resolution technology to reconstruct the volumetric informations inside the brain as well. So we can do these whole brain interactions and take advantage of the EEG's temporal resolution. And really, we're able to go to the operative rooms to record intraoperatively for both the cerebral EEGs in the epidural settings or even in single neuron recording. So as you see, this is probably the very first Purkinje cell that record in human ever. So even we have some discoveries in human, and when we're trying to crack the pathophysiology, inevitably has to go to animal models. So typically we do tissue clearings to understand the anatomy and connections of the brains. And then we do these multitextuals and optotrol recordings in awake behaving mice and simultaneous work with optogenetics in a closed loop settings. And we also do 2-photon kill-cell imagings, and for example, in these freely moving settings, or you can do this sensory triggering to see how the cerebellum cope with the sensory informations, and train the mouse to move at various speed. So do these speed-related recordings, and sometimes we do this closed loop visual motion feedback systems in order to understand how they incorporate external informations. And of course, since we're doing motor kinematics, then we have to leverage deep learning to record this moving trajectories in details, and then do this 3-dimensional trackings of motions. And in terms of technology, because we are doing tremors and about the frequencies, so we are redesigning based on high school physics to redesigning a force play with intrinsic oscillation, so we can get the frequency-based information more precisely. And in terms of kill-cell imaging, we are recording 3D structures and across time, so it's a 4D imaging. So we're using AI-based technology and leveraging the continuity of the anatomy to make it more better signal-to-noise ratio to see these kill-cell dynamics more precisely across time. Okay. So we also do collaborate with other electric engineers to see the vasculature using optic coherence tomography. So making the miniature versions of optic coherence tomography. This OCT can see the vessels and see how the neurodynamics and the fully compatible with other two-photon-based imaging systems. So basically, we are doing this translational neurodynamic research, and we focus on specifically the real-time neuronal activity that directly encode behavior details. And we also do human translation, as you see in the clinical electrophysiology settings. So today I would like to share about our recent discoveries about quantitative codings of motor controls. So as we all know that our emotions are incredibly precise in terms of timing and also in terms of space controls. And one of the most incredible features is their reliability. When you do this hand mountain climbing, you don't want to make even one mistake. And that is how precise our motor system can do. Well, one of the caveats is that everybody's neural network is somehow different. So it seems to be like a personalized motor control should be the solution. And in fact, it is the dominant way to do neuroscience nowadays, to leverage deep learning algorithms, but some sort of giving out some of the understanding of the mechanisms. Where we are more old-fashioned, we would like to know whether there are actually fundamental formulas, or in AI era, it's called domain knowledge. Is there a Newtonian-like principles of motion that has mathematically precise and can be applied to all individuals in the system neuroscience level, in the macroscopic level? So in order to address that, we are taking advantage of me being a movement disorder specialist. So we see a lot of patients with cerebral disorders, and they all have these motor precision problems. But when you categorize them clearly and carefully, then there will be two categories of these motor imprecision problems. The first is tremor. So in this gentleman, he's very hard to put his pen on the paper to do the spirogram. But when you look at him very closely, you'll see that there's a pattern. He shakes his hand about 4 times per second. So it's a 4 hertz tremor he'll develop. So there's a rhythm, and there's excessive rhythm in his movements. On the other hand, we all know that there's a classical prototype of cerebral disorder called ataxia. So it's not only that they lost the motor precision, but they also lost the motor rhythm. So from the disease perspective, we have been taught and learned that it is very likely that the cerebellum actually engage in the frequency coding of movements. But how do we prove that? So we think that we should start with tremor. Why? Because tremor is an involuntary rhythmic movement. There's a rhythm, and the rhythm is fixed. So it will be easier for us to capture all the details of that rhythm and how the cerebellum ensemble that rhythm. So for example, if a patient with tremor has a 7 hertz tremor, let's say 7 hertz. So how is this 7 being generated specifically in the cerebellum? So that is a question about how the cerebellum do the math. So we all know the cerebral anatomy, and we'll focus on the olivocerebellum in today's talk, and then we don't need to spend time on this slide. And then we leverage our previous study that we understand the tremor pathophysiology. So what happened is that both in the mouse model and in human subjects, that a patient with essential tremor will lose their delta-2 proteins in the Purkinje cells. And with this loss, the climbing fiber will overgrowth. And they will not only grow more distally, but they will have a lateral crossing to innervate more Purkinje cell on the lateral side. So they will create a super strong and synchronous oscillations in the cerebellum that will cause tremor. So during the journey to prove this concept, we created a grade 2 animal models with this delta-2 protein loss, and that will generate a 20 hertz tremor. And we also create a cerebellar EEG technology to prove that human cerebella also have these oscillations. So in this study, we are pretty sure that the cerebella actually generate this tremor-dependent oscillation within the olivocerebellum. So the next question is about the frequency. So yeah, how is this frequency being generated in the olivocerebellum? So not surprisingly, we have to go back to the neuronal level to see how the neurons actually encode this frequency. So our guess is very simple and straightforward that, okay, so if the olivocerebellum is doing the frequency generations, if we record one of these 3 olivocerebellar loops, for example, recording the neurons in the DCN, then because the mouse has this 20 hertz tremor, the mouse has faster tremor, and then we should able to record a lot of DCN neurons firing at 20 hertz. So to explain it's 20 hertz tremor, right? So that's what we did. And boom, it doesn't look like that. So as you see that in DCN neurons or Purkinje or IO neurons, all of them are firing from very slow to very fast. There are all sorts of frequencies, but they are very rarely coincide with the tremor frequency. And even their means are away from that frequency. And their bursting rates are lower, so they couldn't explain their their tremor frequency. So it is obvious that the neuronal firing rates are unlikely to explain the frequency of tremor. So next, we're trying to say, okay, maybe it's not the firing rate. It is the tuning rate. So what do I mean the tuning rate? That's that DCN neuron is firing at 40 hertz and going up to 60 hertz and coming back to 38 hertz and coming up to 45 hertz and up and down 20 times per second. Then we got a 20, right? So maybe it's the tuning rate that works. So in order to prove that, we use a mathematical tool called vector strength analysis. Basically, we are just using a sinusoidal waveform as a frequency vectors and trying to use this phase analysis to see whether the timing of the DCN firings can be explained by some of the frequency that will have the base phase bias coincide with the firing time and the firing probabilities. But unfortunately, we didn't see any special frequencies pop up from this. So this is another dead end. And luckily, because we use this multi-tetrode system to record a lot of neurons, we'll start to see something magical. So when you put more and more neurons together and their collective firing probabilities start to converge to a certain frequency. When there are more and more neurons, these convergences are stronger and stronger, and they are clearly converged to the same frequency of their cerebellar oscillations and the same frequency of their tremor. So all of the 3 numerical numbers converge to the same one. And then use this cartoon to explain that each of the neurons may fire stochastically, but when you add them together, you start to see their firing probability has this tuning properties. And this tuning frequencies will be exactly the same frequency of the cerebellar oscillations and the same frequency of the tremor. So we now see how this frequency being computed in the cerebellum. So it actually do the same job within DCN, within IO, and within the population of PC neurons. So they're actually working using the same algorithm about these 3 places, but I just don't have the time to show you all the data. And we can also evaluate this population coding from the other perspectives. So we use the Hilbert-based phases to match from the true motions of their mouse and match their DCN neurons firing time. So each time will give up an instantaneous phase generated from the motion. And you can see that some of the neurons are very phase biased. So they are firing aligned with certain phase of their motions. But in shuffle cases, they are rather evenly distributed in terms of their firing phases. In the population level, you can see that this tremor mouse has a greater chance to have a slightly higher probabilities for the phase tuning. the best probability the phase lag will be, the protein index will be 1, so they are pretty small. But in Purkinje cells, there are some of them has very nice phase tuning. So it is totally possible there are several expert neurons of Purkinje cell that actually dominated the frequency coding, and all the other neurons are just noise. So in order to exclude this idea, then we take away all the good neurons, better tuning neurons, and left all the relatively poor neurons, and do this recruitment again with better strength analysis, and you will see almost identical things. So it is really that the cerebellum use this populational codings. They don't care whether it's good neurons or bad neurons, as long as their collective frequency converge to a strong frequency tuning, and they will generate that corresponding motions. We also trying to understand the interactions across the olivocerebellum, the IO, PC, DCN loops. And we understand that the phase bias between cerebellar Purkinje cells or cerebral cortex versus DCNs are anti-phase, nearly 180 degrees. So to us, it's probably telling us that the PC use GABAergic neurotransmitter to inhibit DCN, so they are likely to have these anti-phase correlations. From IO to the cerebellar cortex, it's a 60-degree lead from the IO perspective. So it's like that they actually coincide with these oscillatory circuitries and the transmission. So with this knowledge, we know that the cerebellum uses populational coding to generate tremor frequencies. But it's an observation. So we would like to understand whether, if it is true, then we can actually create populational firings and make the wild-type mouse become a tremor mouse, right? So in order to do this, and non-surprisingly, we use optogenetics to force the neuron fire. So in this specific case, then we transfect tronodopsin into the DCN neurons and then shine the light at 20 hertz. So when you turn the light on, the neuron will be activated and fire like crazy. It can be up to 30 hertz or even nearly 600 hertz. When you turn the light off, it's probably 40 hertz. So none of them are actually firing at 20 hertz. But since we are shining the light at a lot of neurons, so they will collectively have this very, very nice 20 hertz populational tunings across the population. And with this, we are able to generate, using optogenetics, to generate instantaneous 20 hertz oscillations in the cerebellum and transmit to motor cortex and actually generate the motion. So if you see the video, that the mouse are wild-type mouse, but when you shine the light on, they will instantaneously develop these fast shakings of their head and their limbs. So yes, we are able to make the wild-type mouse become tremor. And because it is important, so we are thinking whether we can modulate the tremor frequencies. So we took advantage of the original mouse model. They have 20 hertz tremor. And we do the same thing, but this time we shine a light at 13 hertz. So when the mouse original has this 20 hertz cerebellar oscillation and 20 hertz tremor, and when you turn on the light, then not only the oscillation is 13 and the motions shift to 13. One of the very important features is that the original 20 hertz tremor and 20 hertz oscillations has been washed out. This tells us that the olivocerebellum actually play a critical role in frequency formation. If you shift the frequency within the olivocerebellum, then first of all, they are generating the new frequency. And a more important thing is that they no longer able to generate the original frequency they need because you just push them to another frequency. So this is key information that we learn from this experiment and see that the olivar cerebellum actually play critical roles in tremor. Okay, so we now know that it's in the animal models, we prove that the populational coding actually can generate tremor. And give you a little bit about the human evidence. So back to 2020, then we use cerebellar EEG technology to prove that as compared to control subjects, the patients with tremor will develop stronger cerebellar oscillations. And this oscillation is approximate at their tremor frequency. And the strengths of these oscillations are actually correlated with their tremor severity. So we know that in patients with tremor, their cerebellum will have these oscillations. Since oscillation is rhythmic, we can play tricks on them. So how do we do that? So we first of all, we record their tremors. So we ask them to stretch their hand, and their hands will shake at a certain frequency and record that frequency. For example, it will be a 4 Hz tremor in these patients. Then we use transcranial alternative current stimulation, TACS. It's a non-invasive current stimulation. So basically, you just put a sponge, put it in the salt water, and put that in the cerebellum. The other reference are in the shoulder, so they'll create a closed loop, and you can inject currents, very, very weak currents. You don't even feel it into the cerebellum. But this time, we inject the current at their tremor frequency. So in this case, if they shake at 4 Hz, then we give them a 4 Hz oscillation. So you'll ask me that why bother to give them the same frequency? So the idea is that we're trying to do the anti-phase stimulation. So if the neuron want to fire, we drag their currents away. When the neuron want to be silent, we push the current in. So because they are at the same reason, so we can constantly do this over time. So by doing this, we actually cancel out their cerebellar oscillations or disturbing them very, very severely. And by doing this, you can see that when we record their tremor during this TACS on time, their tremor frequencies becomes unstable. So they are no longer have this stable tremor across time. And more importantly, because you mess up with the system, so it actually reduce the tremor intensity and become a potential therapeutic strategy to treat tremor patients. So and all this works and are published last year in Science Translational Medicine. So this is about tremor. So we know that cerebellum can encode tremor frequencies. And how about ataxia? They are loss of rhythms, right? So we don't have time to go to detail, but let me use this graphical summary to guide you that we use human pathology to see a lot of ataxia patients. For example, spinal cerebellar ataxia, type 1, type 2, type 6, they are genetic ones. And there are non-genetic disease, for example, like multiple system atrophy, the cerebral type. So this MSAC is the most dominant type of non-genetic cerebral ataxia, and also DRPOA. All of these different genetic or non-genetic common ataxias have one common pathology features is that they will have this climbing fiber regressions in a relatively early stage. So in terms of tremor, there are overgrowth of the climbing fiber. In ataxia, they are actually shrinking of the climbing fiber. And this loss of climbing fiber actually caused their cerebellar oscillations has been lost in both mouse and human EEG. So they lost their necessary cerebellar oscillation during motion. And reflecting to behaviors, they actually lost their required frequencies of their behaviors and cause this dysrhythmic movements, and which one of the definitive features in ataxia. In a therapeutic perspective, that if you use chemogenetic to boost the IO excitability, so the climbing fiber will fire more frequently across their need on demand, you can actually partially improve the motor rhythms and improve the motor function. And it just has been published. If you're interested, you can look at the details about all sorts of different ataxias and how we manage in animal models. Okay, so we now know that in tremor, there are climbing fiber overgrowth and too much rhythm. In ataxia, there are actually loss of rhythm and loss of cerebral oscillations as well. So we're more interested about how about normal motions, right? That is something we really want to know. And then in our daily movements, we can also separate them into a rhythmic movement. For example, like I trying to move my hand rhythmically. Well, it's not really a functional movement, but we can do that, right? So it's a rhythmic movement. We're expecting the cerebellum should generate a fixed frequency over time, right? How about a non-rhythmic movement? Then we put this non-rhythmic movement a little bit later. We'll just explain the rhythmic movement for now. That if we generate a rhythmic movement and the cerebellum is working on that too, then we should see that cerebellum will be oscillating at the frequency of that movement, right? So how do we prove that in animal models? We're trying to ask the mouse to kindly move at the same frequency, but they just don't listen, right? So we trick the mouse by putting them on this earthquaking platform. It's a vibrating platform. So we are creating a sinusoidal oscillations horizontally at the platform. You can see the mouse are standing very nicely and they are compensating their movements. So their head are moving really stable, and then they could easily learn how to do this. So by doing this and recording their canceled oscillation in their head, so we can calculate the compensatory motions. And then we can use a different frequency to see whether their cerebellum can generate the corresponding frequencies. So yes, we can see that the cerebellum can generate the compensatory oscillatory frequency. That frequency will match their motion frequency. So when you do this second by second recordings across different animals and across in different time, they are very precise, and the R square is very close to 1. And if you do it in deep cerebral neurons in the interposed subnuclei of cerebellum, then we do this the same more than 2,000 seconds, and the correlation is 1. So they are very precise. And we also do the EMG recording to make sure that we are not recording something arbitrary. Truly their motions are correlated with their cerebral oscillations. And we're also recording the accelerometer and the vibration to confirm that it is coming from motions, not just the sensory feedback signal. So it is the motion that the cerebellum is generating. Okay. In terms of neuronal level, we do pretty much the same things, that we see that the cerebellum can do population codings to align with the frequency they need. So they can do the frequency on demand, not just the trap frequency of tremor, but the frequency they want. Okay, so we know that the cerebellum can use populational coding to generate a volitional rhythmic movement. But how about non-rhythmic movement? When I try to grab a cup of coffee, it doesn't really look like rhythmic movements, right? Any goal-directed movement are unlikely to be rhythmic. So how about that? But luckily that about 100 years ago, that Hilbert and Fourier has already solved the problems that any non-rhythmic movements can use for, in this case, that Hilbert transformed to streaming the instantaneous frequencies and instantaneous amplitudes to build any finite signals. So for example, in this case, we want to generate a signal like this or a movement like this. So we only need a dynamic change in frequency over time and a change in amplitude over time. Then by streaming these two information, then we can create these changing oscillatory waveforms that are not actually fixed at any frequency. Okay, so in theory that you can use this same algorithm to generate any movement. So the problem becomes whether the cerebellum actually can generate dynamic changing frequencies over time. So to probe this, then we train the mouse to standing on this vibration waveform, but in this time, their vibration frequency are linearly increased across time. So no two time points has the same frequency. The frequency are strictly increasing across time. And the mouse had to learn how to stand on this and move freely on this at their own will. And as you can see that the mouse can do this very well, and they learn how to do this, and their cerebellum actually can generate an increasing frequency over time, and they create, and that's coupled with the corresponding motions. And they are very, very highly correlated across all mice and across all the recordings in different trials. So they can do this very, very precisely. And we want to do something even more natural. So we just put a thirsty mouse in front of the water spout so they could leak and get water. And we let them just leak at their whatever they want and recording their leaking kinematics and then recording the DC neurons. And then what happened is that their leaking frequencies and their cerebral oscillator frequency are aligned pretty well. So even in a very natural leaking movements, they can do this frequency coding pretty nicely. So if the server actually computing these instantaneous frequencies or the dynamic frequencies of motion, then the next question is that, can we actually engineer them to force the mouse to move at the frequency we want against their will? So that is exactly what we do. We use optogenetic to stimulate the DC neurons, again with this chirping waveform, so the frequency become faster and faster and faster and faster. And then what happens is that you can successfully generate the cerebellar oscillations with increasing frequency over time and force them to move at your design frequency. And you calculate the baseline frequency, they are very, very tightly correlated. So in terms of frequency coding, this will be supremely precise. And we can also push, in a single neuron level, you can appreciate that how the DC neurons can adapt very rapidly across different trials, because the interstimulus interval actually changing rapidly across time, but the DC neuron actually can cope with them. So their firing probability actually changing quite nicely across time. So finally, we push them to a more stringent scenario. So we are not using a linear chirp. We are actually using complex chirp to change the frequency very complicatedly across time to mimic even more natural movements. So you can see that the cerebellum actually can cope with this and force the mouse to move at the frequency we designed with precision. Okay, how about human? We cannot do this very, very violent stimulation, but we can do two things. First of all, we can record their cerebellum, right? So we ask them to do the volitional tapping at their free wills, trying to ask them to do 4, 5, 6 hertz and record their muscles and their cerebral oscillations. Then you could see that they can do these pretty nice tappings, and they will generate the corresponding frequencies. And they are pretty precise and across all individuals. Their cerebral oscillatory frequencies and their tapping frequency are matched. Even if they are trying to tapping at 6 hertz, but they're actually tapping at 5, and their cerebral oscillation will be 5. And the second thing is that we can do the same tricks with TACS. So what happens is that if you do the volitional tappings and we do the anti-phase stimulations, and you actually will tapping worse, your frequency will become unstable. But in the reverse way, if our stimulation is aligned with your cerebral oscillations during your tapping at the same frequency, you're actually tapping better in this frequency. You won't know this, but we can record your kinematics and tell you that you're actually doing better. So in summary, that to build a signal of motion, or we can see specifically the muscle contractions, then you can use two parameters to make it happen. So one is instantaneous amplitude, and the second is instantaneous frequency. So in terms of frequency, we figured out that cerebellum can do this very precisely using populational codings, and they can change that with the dynamic coding, so they can change frequency over time. And because it's so precise, and we can use optogenetics to create and force the mouse to move at the frequency we want. And we can prove that the cerebellum also used the similar algorithms in humans by recording them and using TACS to manipulate them. One of the key features is that across all testing mice and all testing humans, these algorithms are identical. You don't need a calibrating factors. The cerebellar frequency over time will match the motion frequency over time. So it's a true Newtonian-like features that we can use for probing motions. And it's just published 3 hours ago. So if you want to see the details, you can look at this paper. Okay, so we know that a cerebellum is computing instantaneous frequency of volitional movements. And if you have climbing fiber overgrowth, you will have too much oscillation and there's a trapped oscillation. So they will have an involuntary rhythmic movement. If you are unable to generate sufficient instantaneous frequencies, then you have ataxia because your climbing fiber is regressed and you will fail to generate sufficient rhythm. Okay, so why is it important? So we can recap the essential principles to compute the volitional movements, to understand how our brain can control our movements. So in fact, you only need three things. First of all, if you do a motion, you need to know which muscles has been activated. So it's an ensemble of the muscle. For example, if I do the reaching movement, my bicep and tricep to activate at different time, but they are in these ensembles. The second thing is the timing. So when's the time that the muscle has to contract? And the third thing is the amplitude. When you need to contract, how strong it should be. So it is actually, if your brain control these three things, you actually control your movement. So by deconstructing this motor control into these three parts, you actually notice that we actually already knew the topography of this ensemble things, right? We have this small humans on our motor cortex or even in our cerebellum and the basal ganglia to tell you which part of the brain is activated during which part of your limbs. So you roughly has this topography already. And the study I'm just describing actually tell you the timing. Although it's in a frequency domain, it's actually tell you how or when is the time that muscle need to be contracted. And so the final piece is actually the neuronal synchrony. We need to know how the neurons forming that muscle contraction amplitudes. If we have all these three things in precision, then we know how our brain control our emotions in that incredibly precision. And before I end my talk, there's one more thing I want to emphasize, that my talk is about this algorithmic quantification. For example, like if you want to, in terms of like launching a rocket, you need to pre-calculate the trajectory so the rocket will go to the right places or the space shuttle will go to the right places. But there are another equally important algorithms in the cerebellum. It is endpoint precision, and you've already heard that Abby's beautiful talks about this. So it is equally important. Now there are actually two different algorithms that are working in the cerebellum just to make your motion go directly to and make it successful. It is pretty much like when you're trying to land your space shuttle in the space station, you need these position creating thrusters to make it so precise that they can duck the gates precisely, so you won't lose in space, right? So these two things are currently also both called motor precision, but they are actually very different part of the mechanisms that we need to understand that they are different, and then we need to study them both in details. And then finally, I'd like to thank all my extraordinary lab members. I'm only here to do the talk when they are the one who make the miracle happens. And that's the end of my today's talk, and happy to take any questions.
Ming-Kai, that was wonderful. Thank you so much. You have so much to teach us. I want to begin by, I guess, asking the question about when you were moving the mice at various frequencies. Did you see any compensation for those frequencies? Because I wasn't sure if it's just a sensory response that you were observing, or was there any, like, for example, was the head stabilized and not moving, or was it just being moved at the frequency of the input?
So what happens is their head are more stabilized. So we're actually calculating the accelerometers and their cerebellar oscillation and calculate their Pearson correlations or their frequency-dependent coherence. They are pretty poor. And it's why they are there. It's because their legs are actually compensating the vibration platform, so their head can be more stable.
I see.
And in terms of other sensory, we also calculate directly for the vibration platform because it is the one who generating that rhythm. So it is the strongest sensory input. And in terms of the cerebellum versus vibration, their coherence are poor as well. Yeah.
Looks like somebody in my lab has a question. Please go ahead.
Hi, yeah, really cool work, and I have a bit of a naive question actually. So for the DCN stimulation work, I actually went to your paper and for a little clarity. So the Thigh-1 mice, it mentions that they transfect the DCN neurons with channel rhodopsin, but it doesn't transfect the PCs with channel rhodopsin. Is there any more specificity within the DCN? Like, does the Thigh-1 marker, does that exclude any types of DCN neurons, or do we know if it also transfects the mossy fibers that go to the DCN? And I'm curious how you might like contextualize that with respect to the Purkinje cell results as well.
Oh, that's a great question. First of all, that the Purkinje cell don't express Thigh-1, so we're lucky that we can directly use Thigh-1 mice to stimulate the DCN. But you're totally right. So we also do a complementary study just using chem kinase 2 as a marker to inject the virus into DCN. So it pretty much only infected the DCN neurons, but not the incoming collateral of mossy fiber or other fibers. But they pretty much give the same results.
Yeah. Okay, and you said there wasn't any subspecificity of other cell types within the DCN. It's just all DCN neurons?
Oh, that's also a good question because that in DCN neurons, they are actually glutamatergic and a few subset of GABAergic neuron. We didn't distinguish them, to be honest. We didn't distinguish them. In the reward experiment, we're actually working on these two type of neurons, how they work. They are working very differently in terms of mechanism. But that's a very good question. We didn't know. So we assume that will be the glutamatergic one because it's a major output, but we don't know that for sure.
All right, thank you very much. Thank you.
Thank you.
I thought the alternating current non-invasive stimulation was really impressive, particularly that last results that you showed when it was anti-correlated with the tremor. Is that something that you find? I guess does the human subject, do they adapt to that after a while, or is it like something that can actually be used as a therapy?
That's a great question. That we don't know that for in the long run, because that we only record that like 90 seconds when you turn on the machine. So it is totally possible they have other plasticity kicking, so they will adapt to the cerebellum's anti-phase stimulation. But that probably require a better strategy to do this in the long run. Yeah.
Wonderful. Question in my lab.
Hi, thank you for such an interesting talk. I had a question about the rhythmic activity you found in the cerebellum in the healthy motor behavior. Do you believe that that is being generated by the cerebellum, or does it reflect rhythmic patterns that are generated in the motor cortex or elsewhere and then arrive at the cerebellum? And if so, I guess what mechanisms do you think the cerebellum might have to generate these rhythms?
Oh, that's also a great question. Um, we are pretty sure that the cerebellum is an organ to generate these linear rhythms. And we also record the other places, for example, in the inferior olivary neurons for the calming cortical fibers. We don't record the mostly fiber or in the granule cell layer because there are too much of the fibers that really make us unable to differentiate them. But at least from the cortical IO projections, we see that they don't carry this frequency-dependent information. It is solely in the cerebellum who is generating this final output. But to be honest, we haven't investigated whether the premotor cortex has already generated rhythm. We think it is unlikely because in other motor studies, or including predominantly in human and mouse as well, that motor cortex has this very strong alpha wave and beta wave in different scenarios. If they are generating this complex motor rhythms, then people should already found it very, very clearly in all the other motor experiments. But at least from other people's study, even from my own PhD works in Parkinson's disease, we don't see that at all. So I assume that the motor cortex will give you more like instructive code, and the granule cell is probably the one who is doing this detailed computation to transform them into the time domain information and project to the Purkinje cell and teach the Purkinje cell when is the time you to fire in a population level. So it can be imprecise in a single cell level, but in a population level, it will be very, very nicely coherent with the motion. Yeah.
Okay, thank you.
Thank you.
Are there any other questions for Ming-Kai? Wonderful. Ming-Kai, thank you so much. It was wonderful, and congratulations on your new Nature Methods paper.
Thank you so much. And it's my great honor to join this conference. Thank you.
See you. Bye-bye.
See you. Bye.